Okay, kidney function. We're going to start at the top with an overview of some more kidney anatomy to get oriented. We're going to start out tracking processes in a frog kidney, so I'm showing you a set of pictures here of the major working components within the frog kidney. So a given working unit within the kidneys is called a nephron. And in situ, nephrons have a really complex folded structure as illustrated over here on the right. However, it's a little hard to track processes in this configuration, so over here on the left is one of these nephrons unfolded. So there are three major functional regions to the nephron. We're going to start out examining Bowman's capsule right here. Then fluids travel through this segment here called the proximal convoluted tubule, or PCT for short. And so the word proximal means closer, and then from there it goes to a very short intermediate segment right here, and then reaches the distal convoluted tubule, or DCT, which then feeds into a collecting tubule and a collecting duct. So now if we go back over to this figure on the right, just to stay oriented, here is Bowman's capsule, and then the proximal convoluted tubule travels up here, adjacent to the collecting duct. And then intermediate portions of this tube are down here, and the early parts of the distal convoluted tubule as well, and then the late distal convoluted tubule travels back up here to the collecting duct. I'm pointing out these spatial arrangements right now because the spatial arrangements do become critical for nephron function later on, as you'll see. But for now, let's go back to the Bowman's capsule where the process of urine formation starts. Okay, so inside of each Bowman's capsule is a set of blood vessels called a glomerulus. So note a couple of things here. There's an afferent vessel here that feeds into the glomerulus. Then there's a whole bunch of branching inside of the glomerulus, and then an efferent vessel that departs. And if we examine a close-up view of this vascular endothelium inside of the glomerulus, here's how that looks. So on the left in this illustration is the blood compartment, and this is all lined by vascular endothelial cells, so those are the cells that generally line the walls of a blood vessel. And then this is an example of a blood vessel that is fenestrated. You might remember me talking about those ages and ages ago, right? It really feels like a lifetime by now. So basically, there are gaps between the vascular endothelial cells. Next to that is a basement membrane layer, which really isn't going to factor in much for us right now, but the next layer of cells does. These cells are called podocytes. Because they have these little projections that look like feet. And it's the podocytes that are really critical for determining what gets filtered out of the blood and into the capsule. So in healthy kidneys, only relatively small molecules can fit between the gaps between podocytes. And molecular charge and shape can also matter, but we're not going to really address that right at this moment. It's important to just be aware that this is a sophisticated filtration system. And so in general, molecules like glucose, and then also inorganic ions, and then urea, and free amino acids can move freely in with the filtered fluid. So the concentrations of these substances in Bowman's capsule out here are going to be the same as the concentrations that are present in the blood plasma. And meanwhile, larger solutes, like blood albumins, and other proteins, won't be able to cross over because they're too big. So in someone who has some form of kidney infection or who's experiencing kidney failure due to something like high blood pressure, large molecules and even red blood cells can start to show up in the primary urine. Okay, so now I want to ask if you remember this diagram for how fluid forces and fluid movement work along a capillary within a capillary bed. So as a reminder, in early parts of the capillary bed over here, there's a hydrostatic pressure gradient, right? And that causes the net movement of fluid out of the capillary and into the interstitial fluids. And we call that process in the kidney the process of ultrafiltration. And here's a look at some of the math across the Bowman's capsule. So the blood pressure in the glomerular capillary is 6.7 kilopascals. And meanwhile, the osmotic pressure in the capsular fluid is negative 3.5 kilopascals. And the hydrostatic pressure in Bowman's capsule is negative 1.9 kilopascals. So what that means is the net force favoring filtration is somewhere in the tune of 1.3 kilopascals out of the glomerulus and into Bowman's capsule. Okay, so how much fluid actually gets filtered via the glomeruli to produce the primary urine? So what's produced here is going to be primary urine. Okay, well, the total human glomerular filtration rate is what's stated in this next slide here. It's 120 mils per minute. What this translates into is that all of the blood plasma in its entirety gets completely filtered every 30 minutes. And I want you to just think about that for a second. So if you drink something such as caffeine, all that caffeine is going to pass through your kidneys within a 30-minute time window. And also think about what a kidney dialysis machine has got to do for someone whose kidneys aren't functioning correctly. Okay, so that's not always the only mechanism of movement at the proximal end of a tubule across all animals. I want to show you one other mechanism broken down step by step. And this mechanism reflects what happens in the insect Malpighian tubules from that video in the excretion lab. So first, let's picture things starting out like on the left here, where there's an even distribution of two solutes, the red and the blue solutes, between the space inside the tubule and the surrounding fluid. So if the cells that line this tubule use active transport to transport in the blue molecule, then suddenly the osmotic pressure inside of the tube is going to go up. And that's going to facilitate the movement of water into the tubule. And that's actually going to push the fluid down the length of the tubules. This is a blind tubule here. So now the concentration of the red solute is lower inside the tubule compared to the outside. So that means that this red molecule can then just simply diffuse into the tubule. And again, that whole process is going to start pushing the solution down the length of the tubule. So this is a second type of mechanism that can sometimes contribute to the movement of solutes in water from the proximal end of a nephron or nephron equivalent further along the length of that blind tube. And there's something else I should point out about that initial filtration step. If we're filtering 120 mils of blood plasma per minute, is that how much we usually pee out? It's really not by a long shot, right? So the next part of the process is going to involve the recovery of fluids and some solutes, plus the secretion of other solutes further down the nephron or tube. And again, as with what we saw for osmoregulatory methods in marine teleost fish guts, this method can facilitate the concentration of certain molecules for excretion while facilitating the reabsorption of other molecules, including water. All we need to do, is understand the spatial arrangement of the relevant transport proteins, and we'll have a reasonably good picture of events. So we're going to take a look at this again in that frog kidney first, because the dynamics are a bit more involved for the mammalian kidney. And once we get the hang of things in the frog kidney, we'll be better set up to consider the mammalian kidney. So we're going to start out here by drawing out a picture of the frog nephron. Okay, so as noted previously, we get primary urine produced via ultrafiltration at Bowman's capsule. Fluid is then going to pass through the proximal convoluted tubule, and then through this intermediate segment to the distal convoluted tubule before heading to the collecting ducts. So what happens to those, the glucose amino acids and ions? So in the proximal convoluted tubule, there are active sodium transporters that are going to transport sodium back out of the urine. And then there are also chloride ion channels. So chloride ions can get reabsorbed passively in association with the movement of sodium. Glucose also gets reabsorbed here. And that happens via a secondary active transport mechanism. And amino acids also get reabsorbed here, and we won't dive into that in great depth. Okay, so what that all means is that by the time the urine reaches the end of the proximal convoluted tubule, it's still isosmotic. To the blood, because there are also aquaporins that are present throughout the proximal convoluted tubule. Right, so those are going to permit free movement of water along with the movement of the solutes. So what that means altogether is that about 20 to 40% of the water that got filtered in via ultrafiltration at Bowman's capsule gets reabsorbed in the proximal convoluted tubule. There's one solute, however, that's not going to return to the interstitial fluid in the kidney, and that solute is urea. So if the urine is isosmotic as it leaves the PCT, what that means is the urea concentration is going to be higher, and it's going to be a larger proportion of the remaining dissolved solutes. Okay, so now we'll move on to the distal convoluted tubule. And here's where things get more interesting, and we have to think about the nephron as a dynamic tissue in two different contexts. First off, though, sodium chloride active reabsorption continues. And so what that means is that the amount of sodium chloride that winds up getting lost in urine is reduced to facilitate maintenance of the internal tissue sodium chloride levels. So then we have to think a little bit more about the water that winds up getting excreted as part of two different fractions. Right, so there's a certain fraction that is required to be excreted to be able to excrete the other dissolved solutes that are in the urine, such as urea. And as a reminder, amphibians have a maximum possible osmotic U to P of 1. For amphibians. So their urine osmotic pressure can't be higher than the plasma osmotic pressure. However, amphibians can also have an osmotic U to P that's much lower than 1 to get rid of extra water. So here's our extra. So the question is, how is that done? We're going to consider this in the context of those two situations of diuresis and antidiuresis again. So during diuresis, we have ample body water, so a frog's looking to get rid of the extra water. So our ADH levels are going to be low. And under those circumstances, the DCT, distal convoluted tubule, has low permeability to water. So what that means is this fluid is just going to travel through the DCT. Some sodium chloride is going to get reabsorbed, and at the end, we wind up with a larger volume of dilute urine in the bladder. So just to reiterate, so here, if the urine is isosmotic as it leaves the PCT, there's going to be further salt reabsorption in the distal convoluted tubule. So altogether, you're going to wind up with urine that is more dilute than the blood fluids are. Okay, so that's under the condition of diuresis. So now we should look at antidiuresis when the frog is looking to retrieve more water. So under these circumstances, the ADH levels are going to be higher. And what happens on a cellular level is this. So let me draw a little DCT cell here. Okay. So during diuresis, the cells along the DCT will have aquaporin proteins that are sequestered in vesicles that are inside of the cell. So, here's my vesicles. Here's my aquaporins, sequestered inside of the cell. And they do actually have aquaporins that are also present along the basolateral membrane. So here's the apical membrane. And so this is where the interior of the DCT is, so where the urine is located, right? So here's blood. Okay. So this is a situation during diuresis. So in contrast, during antidiuresis, when ADH is released, it binds to a G protein coupled receptor here on the basolateral surface, and we get signal transduction to protein kinase A. And then the vesicles that contain these aquaporin proteins get mobilized to the cell surface. And so now if we have aquaporins here, as a reminder, if we've got sodium and chloride transported out, that's going to push the osmotic pressure in this direction, and so then in antidiuresis, with that happening, we can get a retrieval of water from the urea. And so that water will then flow back into the plasma. But I want you to notice, again, with the frog arrangement, that this is only going to allow the production of isosmotic urine. At the very best. So yeah, it's going to contain more urea. And it's going to contain relatively less sodium and chloride, so it's going to facilitate the differential excretion of urea and a urea U to P ratio that's greater than 1. But the frog can't do anything to retrieve any more water than that. So interestingly, it's also possible for many amphibians to use the same trick with the aquaporin proteins in their bladders too. So a lot of amphibians actually treat their bladders like a canteen. When there's plenty of water around, you can produce dilute urine and then store it in the bladder. And then if things dry up, mobilize those aquaporins and osmose that water right back into the plasma. Frogs can also engage in some active sodium chloride transport in the bladder as well when ADH levels are high. And I kind of hope that suddenly you're never going to think about bladders in quite the same way again. So we're going to wrap up this section now by taking a look at a graph of the overall consequences of the spatial arrangement on urine osmotic pressure during diuresis. So this graph here shows the osmotic pressure as a percent deviation from the plasma osmotic pressure for two amphibians, mud puppies and frogs, and it also shows that in the darker blue colors, the chloride ion concentration deviation, just taking chloride as a representative token ion. So note that both water and chloride ions are more or less in balance across the proximal tubule. So this is all hovering, fairly close to zero. However, they drop in parallel across the length of the distal tubule such that the chloride ion concentration is very low by the time urine reaches the ureter due to the passive reabsorption of chloride along with sodium. And in the meantime, there's also a strong osmotic pressure gradient such that a large volume of dilute urine gets excreted. So again, this is under diuresis. Okay, so that's amphibian kidney function. And again, I want you to think about amphibian kidney function, both in terms of understanding how it works in general, but also as a way to set the stage for examining and exploring mammalian kidney function. We're going to see that a small section of the nephron, one that I didn't really talk about here for the amphibians, but this intermediate segment between the proximal and distal convoluted tubules, that works very differently in the mammalian nephron and helps to explain how it is that we can get an osmotic U to P that's greater than 1.